Hello, my name is Albert Keung. I'm a faculty member in the Chemical and Biomolecular Engineering Department at North Carolina State University. I want to thank FAST for their amazing work bringing this community together, and I hope today I can contribute some useful information over the next 20 minutes for the families here. In particular, I'll describe a relatively new class of model system called human cerebral organoids, how they could contribute to finding therapeutics for Angelman syndrome, and also provide an assessment of their current limitations and how we might address them. So first, I won't be showing a lot of data, but I still wanted to put faces to the people doing the actual work in our lab. And this is our research group post-COVID up here on the right. Since March, we of course began wearing face masks in the lab and holding virtual meetings. So in this anxious time, I thought I'd also share a more lighthearted image of one of our Zoom group meetings where we decided to wear a different type of face mask, the hydrating and skin nourishing type. That is, of course, for home use only. And hopefully my group doesn't get mad at me for showing this picture. I'm very lucky to work with this committed group of scientists and engineers. And one strength is the diversity of their backgrounds, coming from engineers, biologists, chemists, and that of our colleagues across the campus and within the FAST community who we draw upon frequently for advice and collaboration. So now this concept of diversity in a somewhat different sense is one of the major motivations to use human cerebral organoids in Angelman syndrome research and development. And it's where I'll start this presentation today, even before I discuss what organoids are. In particular, there are two types of diversity that have important implications for therapeutic development. There's a diversity in the number of cell types, so at the cellular molecular scale, in the human brain. And there's also diversity in the genetic origins of Angelman syndrome. So I'll describe what these two things are. So first, we know that Angelman syndrome primarily affects the brain, and early work identified neurons as the important cell type in particular. However, there are actually many different distinct types of neurons that each release and respond to a host of different signaling molecules, some that tamp down electrochemical activity, others that increase it, for example. Which of these cell types are important in Angelman syndrome? In which parts of the human brain? And when are specific cell types affected or not? So these are important questions for us to understand, not only to understand how things are different in Angelman syndrome, but also potentially to deliver therapeutics to specific cell types or specific brain regions in order to have the greatest and safest benefit. So in addition to the complexity and diversity of the human brain, there is also considerable genetic diversity amongst individuals with Angelman syndrome. Shown here is the region of our genome that is important in Angelman syndrome. The specific names of all of these different genes is not so much important here, but I just want to call your attention to UBE3A. So many of us have focused on the role of this gene here in red, in particular because loss or inactivity of UBE3A leads to Angelman syndrome. However, as you know, the Angelman community is quite diverse with different characteristics and needs. And some of this is due to the fact that Angelman syndrome is actually quite genetically diverse. There are many different what we call genotypes. So in many cases, what is known as a class 2 deletion is where this entire segment of genes is deleted. And this deleted region includes UBE3A, so this is why it leads to Angelman syndrome. But there are also several other genes that are likely to have impacts on the function of neurons that are lost as well. There are also larger class 1 deletions that include these 4 genes here on the left, and these can lead to more severe clinical characteristics. And there are also situations where UBE3A is itself not deleted, but its activity or expression is attenuated in more indirect ways. For example, there are over 30 distinct mutations in the sequence of the UBE3A gene that have been discovered and associated with Angelman syndrome. And there are many other types of mutations or chromosomal duplications that are known to affect the activity or expression of UBE3A as well. So it remains unclear how each of these different situations and genotypes specifically affect neuronal function and neuronal development, and ultimately Angelman families. And furthermore, it's unclear how will current potential therapeutics be, how effective will they be for each of these different genotypes. And finally, are there specific therapeutic options that could be tailored for each of these specific genotypes? So these are challenging questions to answer for several reasons. They're challenging because of the complexity that, of the genetic complexity that there is and the cellular complexity of the human brain. It's challenging to create an experimental model that can reflect even just one genotype. And it is hard to create models that can rapidly and efficiently access the diversity of the human brain. However, I believe we can look at these challenges also as an opportunity to try and identify new therapeutic targets and broaden the impact that science can have on what we can recognize as a diverse community of Angelman individuals. Furthermore, understanding different mechanisms could actually help us understand common mechanisms that impact all Angelman individuals. And if we can develop experimental models to address these types of diversity, we may be able to create more tailored and safer therapeutics, both from a mechanistic standpoint, as well as creating drug screening and testing platforms that better mimic what's happening in the cells of different people with Angelman syndrome. So there are several experimental models that have helped make significant progress in this area. So these include rodents, pigs, non-human primates, and human cell cultures. Today, I'll specifically focus on a new, relatively new system developed over the past decade or so in other contexts, and that have just recently begun being applied to Angelman syndrome. So these model systems are called human cerebral organoids. So these organoids are microtissues about 2 to 5 millimeters in diameter. We grow them as detached floating cultures, and they can be maintained for over a year. Importantly, they are comprised of human cells that reflect human cellular and molecular properties. So if you were to zoom in on one of these, they are essentially microtissues composed of anywhere from 100,000 to over 1 million cells. And just for context, the human brain has over 100 billion cells, so these are nowhere near close to what an actual human brain would be. But they provide a lot of the complexity in cell types that are important to study. So rather than just looking at my illustration here, here's what an organoid actually looks like. If you were to slice one open down the middle and then stain it using stains that will label different cell types different colors. And what is particularly cool is that you can actually see polarized structures, like these layers of different cell types in red and blue and green, that form within these organoids. And these layers actually mimic the layers that form in the outer region of a real human brain. So this is stained image of a human brain. You can see these layers out towards the periphery of the brain. Remarkably, this happens without needing to create any sort of external patterning to force it to happen. This patterning happens on its own. So a key feature of these organoids is that they contain many diverse cell types of the human brain, and so capture this aspect of diversity in an efficient way. Not only that, they can also capture the maturation and development of many of these cell types over time. And so why might this be important? Well, as just one example, a key change that had been observed previously in mice was the movement of UBE3A into the center region of cells, the nucleus. And this might be a hint of which cell types are most affected in Angelman syndrome, and therefore also suggest which cells would be best to target therapeutically. However, in humans, this remains unclear where and when this change in UBE3A localization occurred, primarily because it is difficult to do this work on human tissues. and it's hard to get it and ethically work with human tissues. So in contrast, organoids provide a readily available, ethically available source of microtissues. These organoids can create cell types from all parts of the human brain, like I mentioned. So these are just some staining images from Madeleine Lancaster's paper back in 2013 that shows that these organoids can create cells from different parts of the brain, like the hippocampus, the ventral cortex, choroid plexus, etc. So we use these organoids to ask this question of when UBE3A transitions its localization within specific cell types, and which cell types does this occur. So this is an image of one of these organoids, just stained with different colors. As I mentioned before, there are layers of different cell types. So here you can see two different layers of cells marked by the red color on the inner layer and the pink color here on the outer layer. UBE3A clearly changes from a very diffuse type of localization in the red layer, right, on the inside layer, to being concentrated in the nucleus of the cells in the pink layer. Now, I mentioned two challenges that we face. We just talked about capturing the diverse cell types in the human brain. But there's also the diversity of genetic profiles or genotypes that are each associated with each Angelman syndrome individual. So far, what I've shown you is a neurotypical organoid comprised of cells from just one genetic genotype. So how can we use it to generate specific organoid models of Angelman syndrome and capture this genetic diversity? So a powerful aspect of organoids is that they can be created from cells called pluripotent stem cells, and that are capable of generating all cell types of the adult body. And fortunately, these stem cells can be generated from patients directly. So they can be you can isolate skin or blood cells from an individual, and they can be reprogrammed and converted into stem cells. And these stem cells can then be used to make cerebral organoids. And importantly, because all of these cells were derived from this patient, they will reflect the genotype, whether it's a UBE3A deletion, mutation, imprinting center disorder, or uniparental disomy. These cerebral organoids will reflect that genotype. Now, there are challenges with this approach because cells from person to person can exhibit considerable variability from person to person. And so for experiments, we would prefer to have it create these types of mutations on the same, what we call genetic background. So another approach that has become more feasible recently with developments in human genome engineering technologies is to take a stem cell, apply these genome engineering technologies, and where you actually edit the DNA within the cell. And these edits can then mimic a deletion, mutation, ICD, or UPD. And then you would take these cells and then create cerebral organoids, and then they would hopefully capture these these genotypes. So we and others have now generated organoids from either patient-derived or genetically engineered cells, and shown that they can be useful in understanding the functional impacts of Angelman deletions. And so, for example, Stormy Chamberlain, Mark Lalonde were pioneers in these efforts. They had derived stem cells from an Angelman individual with a class 2 deletion. So they were missing this region of the maternal chromosome. So we used these cells to generate organoids and found that they showed when and in which cell types UBE3A was absent or silenced. So for example, these images down here show two different cell types in pink and in red. And it's apparent that UBE3A that's in green is present wherever the pink is, but is absent inside the red cells. So these are the cells that are most likely impacted by Angelman individuals. and these are neurons. So these organoids may be useful in other ways as well, since they exhibit the expected silencing of UBE3A, this absence of UBE3A in certain cell types. They could potentially be useful in preclinical drug testing. So one of the leading therapeutic strategies is to reactivate the paternal copy of UBE3A that Angelman individuals have, but that is silenced. So we used one of the first compounds that was discovered to reactivate UBE3A, indotecan, down here on the bottom, and added it to the Angelman organoids, and saw that indeed UBE3A, shown here in red, was reactivated upon addition of indotecan. This reactivation of UBE3A also matched a rescue in the neuronal activity of cells in the organoids, so the electrochemical firing of these neurons. So for example, on the left are neurotypical organoids, the center are Angelman organoids, and you can see that these neurons are firing more frequently. And on the right, this firing is reduced close to neurotypical levels, and this is where you add indotecan to the Angelman organoids. So this was done using patient-derived cells. However, genetically engineered cells can also be used to generate organoids, as the Sun and J groups showed, with similar electrophysiological properties that we observed, where firing rates were increased when UBE3A was deleted here on the right. So the right is where UBE3A is absent, and the left is where is a neurotypical organoid. So these are exciting developments showing that organoids could serve as powerful experimental models that reflect human genetics. They can also address the two challenges I outlined at the beginning of this presentation. They can capture the diverse cell types of the human brain, the diverse genotypes of Angelman syndrome, and they can also capture genetic and functional responses to putative therapeutics. And finally, they can also be ethically and efficiently grown in cell cultures. So our hope and vision is that we can generate organoids from a variety of cell lines with genotypes that not only reflect the diversity of Angelman individuals, but ask how each individual missing gene, so for example, class 1 deletions would have 4 extra genes that are missing compared to class 2. And we want to ask how these missing genes affect the biology of the organoid. So essentially, the hope is for the research and preclinical community to have a comprehensive set of cell lines for use in research and therapeutic development. And these could be used to ask tailored patient-specific questions, such as, will a specific drug have different benefits or effects in individuals with class 2 deletion versus a specific type of UBE3A mutation. However, while I mentioned that the different Angelman genotypes can be generated, the process to do this remains difficult due to the inefficiency of the genome editing process. And our goal over the next year plus, with the support of FAST, is to develop a genome editing platform that can hopefully help reduce this barrier for academic and industry researchers. So we are testing a combination of molecular technologies, including CRISPR, what are called CRISPR and recombinase-mediated genome editing, and hope to be able to share positive progress on this soon. The goal here is to develop cell lines that any researcher could easily edit to reflect a specific genotype that they wish to study or test therapeutics against. And we are initially developing systems for large deletions and UBE3A mutations, but are also thinking hard about methods to generate cell lines for ICD and UPD. And ultimately, the hope is to have therapeutic modules for all Angelman genotypes. So lastly, I should mention that organoids do have their limitations, as all experimental models do. So for example, currently organoids generate cell types that do not seem to mature past prenatal stages. So we're working on ways in our lab to think about how we could accelerate the maturation of these organoids. Organoids also exhibit molecular signatures of stress from just being in a cell culture rather than being in a natural brain tissue environment. And so there are many groups that are working on this. this issue. And there's a number of other improvements that could make this model better. So improving the tissue organization, the layering structures, different regions of the brain, improving vascularization, so introducing blood vessels into these systems. And of course, all the time, we want to keep an eye on bioethical considerations. So as we make these organoids better and better, will there be a time in which we should stop this work because the organoids might be too close to the real thing? And then we're also working on high throughput methods to analyze these organoids since they're so complex, and so we're working on methods to do that. And then, of course, the one major limitation that will probably always be there is that organoids cannot capture behavior. So unlike rodent models or non-human primates, organoids are in a dish, and so we cannot capture behavior. And so this I think, it just illustrates the importance of having many complementary different model systems. So I'll end by thanking you for your time and your attention, and by thanking FAST for supporting our work. And of course, thanking the students and postdocs in our group that have done a lot of the work in our lab. And I also wanted to show my appreciation for the highly collaborative scientific community that has helped us in numerous ways along the way, providing samples, advice, scientific insights, with many of the names listed here. And I would be more than happy to answer any questions you might have. Just drop me an email if you have any questions. So with that, thank you very much.